All right, guys. So welcome to the day 3 of the workshop. So we just same as yesterday. We just need to download these files to the Hoffman 2 before we begin. So let's go ahead. Get connected to the Hoffman 2. I'm just gonna create a separate directory. Oh, don't forget to activate your Conda environment before you proceed. Activate QCB and then just copy and paste these commands to download material. Here's the snip tree from yesterday. Some genes we need for today, and yep.
We download all of these into Plotly, right?
Yeah, except for the very last line here. You just download it to your local computer.
Sure.
Oh, sorry. I think I gave you guys wrong file for this one. Let me just share it again. For the very last one. Let me just share the new one. You guys are not in the Zoom right now, right?
No.
Let me just share it through the email then. All right, I just sent the email. You guys can download it to your local computer. Yeah, so for today's workshop, we're gonna annotate the genome that we assembled from day 1. And then we're gonna try to look for the core and pen genome from that, using the genome from day 1 and also some other genomes. And then after that, we're gonna try to do some comparative analysis, and lastly, we're gonna visualize some of those results. So before we begin, I just want you to install this program called RORI, which is for the core and pen genome calculation. So the installation time for this one is pretty long, so I want you to just type this in before we begin anything. So because it creates a conflict with the existing environment, I'm just gonna create a new environment with the name RORI, and then this entire command is gonna install the program. And then you can, once you hit that command, it's gonna take a while to install. So you just need to open another terminal and get connected to the whole command too. So we can proceed for other stuff. Yeah. So we're gonna install another program called Proca, which is for the genome annotation. So let's copy and paste that. It's also gonna create a separate environment since it creates a conflict with the existing QCV environment. So just copy and paste that. Yeah, so mine is already installed, so I'm not gonna install it, reinstall it. But you guys go ahead and install it.
Why does it create a conflict?
Because it uses a different versions of like libraries, prerequisite libraries from other softwares we installed a few days ago. Yeah. So Proca is, as I said, is an annotation pipeline. So it mainly annotates the genes in the genomes. Not only the coding sequence, like protein encoding sequences, but also some of the functional RNAs, such as tRNA, rRNA, and non-coding RNAs. Is using so the stuff in the green box here represent the program it incorporates, and the cylinder here represent the database they use. So it is like a multiple it is like a package of multiple programs that are running through the Proca.
So it aligns your FASTA to like it uses those programs to align your FASTA to these types of genomic data. Right?
Yeah, yeah. So first it uses Protigol, for example. So this one extracts the open reading frame in your FASTA files, which is like this predicts the coding sequence in your genome, and then it tries to use a program like BLAST or HMM, hidden Markov model, to annotate them, like to see like which genes they are. Don't forget to hit yes.
I'm trying to download the one on the first download data slide that you said to download to your local computer, but when I click on that drive link, it just takes me to like it says that the file is in the owner's trash and it won't let me download it.
Really? The link I sent you through the email?
Oh. I will use that one.
Yeah, yeah.
Sorry, thanks.
Yeah, so don't forget to hit yes on Rory installation terminal. Just let me know if you guys have installed the Proca in another terminal. If it's ready, we can proceed.
Mine's still in progress. So.
Okay.
Yeah, is Rory installation supposed to take a while?
Yeah, it's gonna
Okay,
take a
yeah,
while.
mine is still running.
Yeah, so I want you to open another terminal.
Oh, yeah, sorry.
Yeah.
Yeah, they're both in progress right now.
Yeah, the Rory like installation takes like really long time. I think it took me like 30 minutes, more than 30 minutes yesterday when I tried it.
Yeah, no, I just opened a new one for
Yeah.
Okay, my Proca is done. Should I switch to the environment there?
Yeah, so you can activate, conda activate Proca after that. Yeah, so you also need the Hoffman to the submit script that we used in day one. So you might want to copy that from day one directory. So I think I have it. I'm just going to copy that to A3 directory. Let's open that up. And yeah, you can give it a 1 hour of running time, and then 2 gigs of RAM and 8 CPU power. And then make sure you change the environment to Proca, as well as the name of the script, Proca. And then copy this command. Yeah, so note that I'm determining, I'm setting up the parameter for all the taxonomic levels so that we can minimize the search space for the database search. But you can just delete this, but it's still gonna work. But for the sake of time, it's always better to designate the right taxonomy. Okay, so once you have that, so our output will be generated in this folder called proca out with the file name qcb.vc. So we're putting in our Illumina assembled data from day one here. So let's just save that and let's submit the script.
What were the parameters of QSub here?
Here. Can you see it? It's a 1 hour running time,
1 hour,
2 gigs of RAM.
8 cores.
Yeah, 8 cores. And once you have done that, you can submit the script, the grade
And,
engine.
the script is just what's on the slide, right? Besides all the other stuff.
Yeah.
And the Proca.
Is yours still installing? Jenny?
I think Rory is done.
Already?
Um,
That was fast. Oh yeah, it's done on my part too.
and then Proca is almost finished.
Okay.
And then I just edited the Hoffman submit script. So I'll submit it.
All right.
Okay, I'll submit my script.
Nice.
And yeah, my goal is finished as well.
Okay. It shouldn't take long to finish the process for Proca.
I'm still stuck in the queue. So.
Okay. Is it running out there first?
My Q stat still says it's in queue.
Oh, really?
So might be because I used a lot of resources last night. So
Oh, you did?
I-
Yours? Okay. Did it generate the output?
Prompted out.
Okay. Do you have something similar to this output in that directory? Broke out.
Similar.
Okay.
I don't have the error summary.
Okay. I mean, mine is still calculating for some reason. So it's like.
Could you remind me what this command is to submit the script?
Q sub.
And then the name of the, thank you.
Is this what you're asking?
Yes, thanks.
Yeah, mine's still in queue. Is it possible to lower the memory usage or something?
Yeah, I mean you can try that. You can just change the numbers if you want to lower the number, but it's gonna slow down the
I'll
process
just-
a bit.
It's already in queue. I'm just gonna leave it.
Yeah.
Yeah, I don't know how often that's to allocate the resources and then just.
Yeah. It also like depends on like how much load it got at the moment, right?
Yeah,
When you submitted it. If people draw like a lot of scripts, then maybe I can share the result file.
Yeah,
with you, so you can proceed. Yeah.
Because I've been in queue for 5 minutes, so
Okay.
I don't know what's going on here. But sometimes, yeah, I've had experience where Kotlin just takes an hour to start running the program or whatever.
Can you just join the Zoom so I can share the link through the
Yeah,
chat?
sure. Is it the same Zoom link as yesterday?
Yeah.
Okay, I'm in the.
All right. Let me just put it up on the Google Drive. Yeah, this should download the zip file. 12.2.
And does this need to stay on Kotlin or is is local fine.
Oh, you need to download it to Hoffman to proceed to the next one. Yeah, you just need to copy and paste that. Yeah, so for those of you who are still running the program, you can also download that output files so that we can proceed to the next step. So Proca will generate this annotation results in various different file format. For example, the FAA contains only the coding sequence in the amino acid format, and FNA file contains the DNA of that protein sequence, and then GFF is a format, basically one of the standard format for the genome annotation, as well as the GBK file, which is also called the gene bank format. So if you take a look at their formats real quick, just go to their directory. So as you can see, we did pull out all the CDS. So this is a FAA file, so which contains the protein sequence. That's why we see the amino acids sequence here. So you can see this gene represent the hypothetical protein, and the ones that has an annotation, a specific annotation, it has a name on it at the title section. So we're gonna use this FAA, the FASTA file that contains the protein sequence. We're gonna use this. Amen. So we're gonna use this for the next one, where we're gonna do the blast search against the set of genes in the genome. So BLAST stands for the basic local alignment search. It is a heuristic algorithm, meaning that it emphasizes on the speed of the search over the accuracy of the results. But it does fairly good job. So what it does is it uses a small fragment of your query sequence, and then it start the search by looking at the exact match of the word in the database. So computationally, exact match takes a less resource than the finding of similar matches. So once it finds the exact match in the database sequence, it then extends their search with the other fragments. So that's how they are speeding up the search time here. So there are different types of BLAST available. Depends on the your query sequence or the database. So if you're searching DNA against the DNA database, you can simply use the BLAST N. And if you're comparing protein to protein, you can use BLAST P. You can also use DNA towards the protein database, where in this case, the BLAST X will try to query all possible 6 frames in the DNA sequence to translate that into protein and then search it against the protein database. T+N for the proteins query against the DNA database, and lastly T+X for the protein encoded DNA against protein encoded DNA. So this for T+X, this is gonna take the longest time because it has to consider all 6 by 6 possible frames, both in the query and the database. And before you do any type of search through the class, you first need to index the database. So this is how you do it. So we're gonna use our protein annotation from Proca result, which is a .faa file here. So we're gonna use that as a database. So let's go ahead and type in.
So should we have, when the proca command is done, like seen a new directory called proca out?
Yeah.
I don't see that. I think that there was something wrong with my script.
Okay. Maybe you can share your screen. Can I see your script?
Sure. Can you enable screen sharing?
Sorry. Go ahead. Is your job finished?
Yeah.
Okay.
I can show you the job log though. It's full of errors.
Um, Let me ask. Yeah.
Is it working for you?
I don't think it's working.
Yeah, okay, I have to enable it. Sorry. Okay, now if you try to.
Okay.
Yeah. Oh, is that what the problem was?
What's the job? Let's see. Oh.
Did you change anything about the script?
No, not really. I'm
Okay.
just trying to see.
Will it make a difference that I was in the wrong conda environment?
I don't think it should because we're activating another environment in the script, right?
Right.
I don't understand why you're getting this error though. I mean, it seems like it's trying to relog into Hoffman 2 for some reason. Bash. Can you change to your email address?
Yeah. Can I do that?
You gotta press I when you, yeah. Okay.
So start with like day one, download everything.
Yeah. So you got access to Hoffman too now? Cool. Or you can just start downloading day 3 material just for today.
So now,
Yeah. Let's just try this interactive environment. 2G RT. All right. Can you copy this?
Yeah.
You got it?
Yeah.
Okay. Let's give it a try. Yeah, since it's working now. So you can just open a separate terminal while it's running,
Okay.
and you can also download the output file just to proceed. So I shared the output file in the chat box. You can just paste this command to your Hoffman 2, the separate terminal, and then unzip it.
Okay, thank you for helping fix that. So you just like requested a computing node and that fixed the problem?
Yeah, so for some reason your script is not running properly in the Hoffman 2, so I just opened up interactive session.
Oh, and then you just did the command from there instead
Yeah.
of in the script. Okay. Is there a reason we're not just doing that in the first place?
Because if everyone's requesting an interactive session, there's last time I ran this workshop, some people couldn't get the node.
Yeah, I had that trouble last time. Okay.
Yeah.
Thank you.
Yep. All right, so back to last. So once you have the PROCA output, you can use this the protein sequence. You can use it as a database for our blast search here. So make sure you activate the PROCA environment since it contains the blast.
so.
Oh, you don't have to install it.
Oh,
If you have a
sorry.
PROCA,
It's
yeah.
part of it, okay.
because if you look at the pipeline, it has a blast in it. So
And the input is the that one.
was it?
How do I do it?
Yeah, this is gonna be the database for our search. So we're gonna make blast db and then dash int, and we're gonna input our the protein coding sequence and the database type. Since it's a protein, we're just gonna write down prot. And if you've done everything correctly, you should see something similar to this. and it should generate the indexed files, something ending with PHR, and then so what we are going to do now is we're gonna try to search for this CTXA gene against our the protein coding sequence. So CTXA stands for the cholera toxin A gene. So I picked that a database. It has so it has a bunch of the toxins from multiple pathogens. So if you go to the Vibrio genus in this database, you will see the number of toxins that they contains, and I just picked the one that are, I think it's this one, yeah. Yeah, so I just picked the cholera toxin A from this database. So we're gonna search this specific gene, make sure that if this gene is in our genome. So let's try to search that using BLAST. So you can copy this to your Hoffman 2 script. You can delete the proc command for now. Change the name. I don't think you need a 8 CPUs for this search. And then submit the script.
What were the parameters here? Is it still 2 gigs, 8 cores?
No, just 1 core and 2 gigs of memory.
Okay.
Oh, sorry, I think the command has an error. Need to fix. Oh, sorry. There shouldn't be a out. So you need to delete this. And submit.
And what was the error that you?
Oh. So there was another out there. We shouldn't be there. So you need to delete it.
Did we need to do the step where we make a blast database?
No, you can just run it on the login server since it
Okay.
only takes like a second to run it.
Okay.
Yeah, so this should be done fairly quickly. So you should have this blast.out file if you've done everything correctly, which will contain this tab delimited files. And this is the header for this file. So QSeq ID stands for the Corey sequence ID. So CTXA is what we have used for the Corey sequence, and this is the target sequence we found in the database. And then the next one is the percent identity. So obviously the one on the top here had 100% identity with the CTXA gene. So there's no doubt that we have cholera toxin A gene in our genome. And the next one represents the length of the sequence, which is about 258 amino acid sequence. So it represents the alignment length, so the one that has a match. And the next one, it represents the mismatch, and then the location of the amino acid sequence. The last two here represent some statistical value. This score, you can think of it as a sub-score. The higher, the better. E value stands for the expected value. So it's like when you open a database, let's say if you pick up the random sequence, what are the odds of that sequence being CTXA? So for the E value, the lower, the better. So for our case, the E value was 0. So obviously it's the lowest possible value. So with the 100% identity and 0 E value and relatively high bit score, we can say that the CTXA indeed exists in our genome. Okay. All right, so next thing we're gonna do is we're gonna try to look for the antibiotic resistance genes.
Should we install this in Roca image?
Yeah, it should be fine. Yeah, you can install it on top of Proca environment. So go ahead and copy and paste this.
I think there's some weird issue with Hoffman because my last man ran instantly, but progress still stuck in the queue. So
Oh yeah, it's still on wait.
I'll probably just kill the job since you're getting that file.
Daniel, I'm trying to run the search the query sequence against the database command, and it says no index file found for the Proca out QCBVC FAA.
Did you do this? Did you make BLASTDB?
Oh no, that was the step I was asking if we needed.
Oh yeah, you need to. sorry.
Okay. So do things get typed between those?
Yeah, so you need to change this db.fasta to this one.
Okay.
CV. And the db type, this contains a protein sequence, so it should be prot.
Okay, thank you.
If it was nucleotide, you should type this one in your CL for nucleotide. All right. Yeah, so let me know if you guys installed the AMR Finder.
Mine is done.
Okay. Okay. Yeah, so similar to Proca, so AMR Finder comprise different pipelines when it comes to searching AMR genes. So it has their own database incorporated into it. So they use BLAST-P, which we just used, and another tool called HMMER, which uses the machine learning technique called hidden Markov model to search the AMR genes. When you first run AMR Finder, you need to download the database. So you can download the database with this command. So go ahead and hit this command. AMR Finder update.
Run the
Yeah.
command directly, or do we need a script?
Yeah, you can. You're not supposed to run stuff in the login node, so try to submit it through the Hoffman to submit script. So here you're putting in the protein sequence as a query. Since we have the entire set of AMRs in the database. So it's kind of reversed from what we did from the BLAST search, because in the BLAST search we used the protein sequence as a database. But anyway, make sure you guys copy this command and put it in the Hoffman script.
Same one for
Yeah. Should be safe. Yeah, so this should finish fairly quickly. You should get this AMR out.txt file as a result. So this is also the tab delimited file. So it's kind of hard to see here, but as you can see, we found these two genes, VARG and CAT-B9, which is responsible for antibiotic resistance against beta-lactamine and the phenyl. Yeah, and they do have fairly high coverage and identity. I think is this for identity? Yeah, it's much easier to read this output file when you like paste it to Excel. But anyway, so yeah, so this is how you find the AMR genes.
Wait, so can you repeat what exactly the difference between AMR and the Flash P is? Because it looks, it's pretty similar. They're both doing like local alignments, right? But like,
Yeah. The stuff they're doing is pretty similar, but here we are using different database, I would say.
okay, so that's
Yeah, so here we are more focusing on the toxin genes. And here we are looking for specific genes that invoke the antibiotic
I
resistance.
see.
Yeah.
Okay, that makes sense. Yeah, so it's just comparing it against two different databases.
Yeah.
I'm not seeing the AMR output file.
Is
But
yours still running or is it finished?
It says it completed like in my email, but
Okay.
I do have like a proca out 2 zip file now.
Yeah,
but I
that's the one I sent you through the chat actually.
Oh, okay.
Downloaded.
So maybe I should just wait a little bit more for the
Can
AMR.
you open your job log just to make sure that if the issues are same as before?
Yeah, right.
Can you share your screen? Probably the biggest number is the most recent one. So 709 maybe.
I think it's this one.
Please do not compute. Yeah, I don't understand why your script is trying to re-log into Wolf12 login server.
Maybe when I like copy pasted the script, something went wrong.
Maybe. Can you re-download the script? Let me just reshare through the chat. 1 second. Can you copy and paste this?
Yeah. Okay, now I really don't know. It's telling me that the command G down is not found.
Was it? Oh, because you're not on the QCB environment.
Mm,
We installed the G down on the QCB environment, if you remember.
yeah.
So on the activate QCB.
Okay, and now I can paste that in.
And can you change the content of the script? So everything should be the same except for the, did you get the PROCA results?
Yeah.
The one we ran with the interactive section? Okay. So you got this directory, right? The PROCA out
Yeah.
and qcb.vc.faa.
Mm-hmm.
Okay. Yeah, so you should be good to go. Submit this. Yeah, seems like you got it now.
Oh yeah, there's the AMR.
Oh, it's still running, I guess. Just why it's empty. Yeah,
Thank you.
you should get it. Yeah. So before we proceed, do you guys want to take a break or?
Sure, yeah.
Yeah, let's take a 5 then. So we got, so far we've done the blast search for toxin genes and the AMR gene search. So right now let's try to do the comparative genomics. So why should we compare genomes? So by comparing multiple genomes, we can know more about the specific organisms. So you can better determine what kind of factors make up the pathogen. So what type of genes actually invoking the pathogenicity of the certain pathogens, or you can even use this technique to find which genes are actually responsible for bioremediation functions in bacteria. And so overall, all these comparative genomics, the goal for a comparative genomics is to understand the evolution better. So when you do comparative genomics, there are a few terminologies you need to know. So homolog is an umbrella term for orthologs and paralogs. So orthologs is the genes that share the same ancestor, which means the genes that share pretty much similar functions. Wherein the paralogs represent the gene copies that are created by a gene duplication event. So in terms of functions, paralogs might have different functions, even though they are exact copies of duplication, because they're prone to mutation when the duplication event happens. So people believe that most of the evolutionary mutational event happens during the paralogs. So when it comes to comparing multiple genomes, we want to specifically find these orthologs, the group of genes that share similar functions. So computationally, the orthologs are calculated based on the reciprocal blast search. If you remember from yesterday, we have mentioned the ortho ANI, which had a similar algorithm as this. Yes, ANI used the fragment of the genome, but here we are using only the coding sequence because we are interested in finding the genes that share the same functions. So we do the bidirectional blast search against each genomes here, and then for the genes that has a top hit to each other, the black arrows here, we can group them together based on their blast results. So if we have so let's say we compared these three genomes. If we found the specific genes that are shared among these three genomes, we call it coorgenes. Whereas if certain genes are not shared and present uniquely in genome B, we can call them accessory genes. So the combination of coorgenes and accessory genes, you can call them pangenome, which represent the entire repertoire of genes in a given set of genomes. So for this figure, the entire set of blocks here represent the pangenome of the genome, of the pangenome of just this given set of genomes. And so the RORI is the pipeline that does the bidirectional blast search with the given set of genomes that we put in. It does have some extra filtering steps to bring down the search space. So it first clusters the CDS, make sure there is no redundancy within a genome. And then it does this the reciprocal blast, which is represented here as a all against all blast search. And then the results get stored in the the Excel format. So you should be able to run RORI with the following command. You should already have installed the RORI in the beginning of this workshop. In order to run RORI, you need to submit multiple GFF file, which is an annotation file that contains the coding sequence, which we just generated for our genome. So in the proca output directory, it should have the output formatted in GFF format, this one. So we're gonna use that, and we're also gonna use the RORI input GFF file. So let's just go ahead and unzip it. let's just create a separate directory for now for Rory. And let's move that stuff into this directory. And then unzip it. I'm just gonna delete the zip file after the extraction. So these are the multiple cholerae genomes from different time points with in the GFF format. So this contains all the CDS from different genomes. So we're gonna copy our genome into this directory. So let's copy our proca output UCBC GFF into the Rory directory. Yeah, so if you've done everything correctly, you should have this GFF file in this directory. And then you can go up. You can go back to day 3 directory and you can now change your Hoffman 2 script. Let's copy the command and make sure you change the environment to Rory. And then for the CPU power, let's give it a like 8 core. Or P shared, let's give it 8. And the running time should be less than an hour, and the memory 2 gigs. Okay, sounds good.
So it's 8 cores, right?
Yeah. And the name should be also changed. So this command will generate the output in the Rory_out directory. Good, so let's go ahead and submit the script. Yeah, so while this is running, I guess we can generate this surplus plot. So this one you don't need a Hoffman 2, you just need to run it on your local computer. So you can download this tool by clicking this link here, the brick.sourceforge.net. You can go ahead and download the brick to your local computer.
We aren't getting an output from that Rory command, are we?
Yeah, we're still waiting for it to run. Mine is still in queue.
Yeah.
I'm not sure if yours are running.
Mine says complete.
Complete already? Okay. Did you get the output? Or
What should the output be?
it should generate the
Yeah,
direct
no.
folder named Rory out.
No, I don't have that.
No? Can I see your screen?
Sure.
Okay, so it seems like you.
I think it's fine. Like you said, it's probably still in queue. I was just wondering if there was going to be an output because I didn't see in the command. Like,
There's going to be.
okay.
Somehow it got closed. Oh, sorry.
It's
It's
it
this
ends in
one, right?
288, 4288.
Yeah, or. 4288. Oh, Rory not found.
That's why it completed so fast.
Mine is still stuck in queue. Is it okay if I lower the parameters to 4 cores?
Yeah. So just your problem is you didn't change the environment. You need to change to Rory. Because we created a separate environment for Rory at very first, at the beginning of this workshop, actually. We can also change the or. All right.
Thanks.
Okay.
Yeah, I changed it before and it started running immediately. So,
Okay.
and it stopped immediately, so something is probably wrong.
Something went wrong. Yours.
Oh, I got the Perl issue. Should I export Perl within the Rory script or?
Did it stop running after the Perl issue?
Yeah, it did.
Yeah, you should paste that.
Paste that into the script.
Yeah, before the Rory comment. Oh, it's just why. Yes, seems like it's running now.
Awesome. Thank you.
Yeah, so minor fix here. The input folder is we named it Rory, so make sure you delete the underbar input there. Okay. Did you say 4.4 worked
Yeah,
instantly?
4.4 ran.
Okay.
Started pretty much instantly.
Anyway, so while we are waiting to finish that running, let's open up the BRIC, which stands for the blast ring image generator. You guys, you can go ahead, unzip the file in your local computer. And you guys should also download the input files from this link here. I think you already have.
Oh, yeah, we already have.
Okay.
You sent them to us maybe now.
Yeah, that's right. Sorry, for some reason my computer is super slow today. Yeah, so you guys should have you only need these two files, two GBK files, and also you need to download the GBK file from our PROCA results so that we can compare our genome against these two genomes. All right, man. See you later.
There's a GBK file in the program.
Yeah. So if you go to the PROCA output directory,
Not sure I see one.
GB. Oh.
Maybe it's in another format, similar.
Actually, it should generate. One sec. Yeah, for some reason it didn't generate it. Let me just send it to you guys.
Sure.
When we unzip the Proca out 2 and the brig that we just downloaded, does it matter which directory we put that stuff in?
I don't think you need a proca out to zip file for this one, for the break.
Okay.
So go ahead and download this GenBank file. To your local computer. And put it under the same directory as this, where these two files are.
So sorry, where are those two files? Are they in the brig?
Yeah, the brig input.
Okay, got it.
So I did a comparison. It looks like GBK file in the zip that you gave us is identical to the GBK file. Sorry, the GBF file. Is it?
GBF.
Yeah. I ran a diff and it's GBK. I was just curious because they had the exact same file size.
Oh, okay. Maybe they changed the
Changed the
extension
don't
name.
have that much.
Oh, thanks. So it's GBF.
Yeah, it's GBF.
Oh yeah. This is the GenBank format. Yeah, but anyway, yeah. So you can use either one, either GBF or GBK. You can use that for brig input. So let's open up the brig. Where's the source brig? So if you can just open up the jar file, just like double clicking it. If you have already installed the Java from yesterday, Jess, you might have a problem opening it if you haven't installed the Java. Not sure. Can you like double click on?
So I'm still having trouble finding the like Korea and what is it, Bangladesh
Oh,
files. That should be if I unzip the brig thing that I download and then
no. I shared that. I shared it to the email.
oh, that was what you emailed us?
Yeah.
Okay.
Because the link on the PowerPoint was in the trash for some reason. Did you get it to open, David?
Yeah.
How about you, Jenny?
What?
No, yeah, blast ring image generator. You can download it. Did you download it?
I did download it. I'm opening it right now. I think. Do I have to open it the same way we open it?
Oh, with the Mac? Oh yeah, maybe.
Yeah.
Yeah, you should control and
Because
click.
I did do that, but it's saying the Java jar file break.jar could not be launched. So I have it here. And I have it right here.
Problem is. Like, on the terminal. See the download to this file.
So every time I use.
For this one, I guess, yeah, make sure you save that. Yeah, save that.
So Jessica,
Yeah.
you need to download Java to run this. We have already installed it from yesterday's material, but you can just quickly download it, I think.
Okay.
Let me just share the link.
Thanks.
Yeah, so can you share your screen? I can help you with the install.
Yeah. Should I just like share my desktop?
Yeah,
Okay.
nope, not this one. Oh. Can you actually copy the link I shared through the chat?
Yeah. I think. No, I don't know if I can see the chat right now.
Maybe. Wait, actually. Let's type that.
Oh, yes, I can. I found it. Here.
Oh, yeah. You can browse down. Choose Java 8. Let's open that up. Can you type in your password? Yeah, so you only need to install it once. You don't need to repeat this process for your homework.
Cool. Congratulations.
To trash. And now you can good.
So, I mean, since you have it anyway, the other thing I'm having trouble with is finding that, like, putting the, I can't get the brig input files in my terminal, like in the directory I'm putting them in. I tried to do it through FileZilla and I don't think it worked.
You can find what?
The Bangladesh and South Korea
Oh, okay.
files. Like that brig input is, that's the wrong inbox
Oh, sorry.
here. If we do this.
Oh, yeah. Yeah, you have it here.
Right, so if I drag those to here, it should just put them in my workshop 15, right?
No, you don't have to. You don't have to transfer it to.
Oh,
like-
okay, I just need them.
Yeah, you just need
Okay.
it on your local computer. You also need to download
sorry.
additional GenBank file in the chat. Put it in the same directory as where the Korea GBK file is.
Oh, this is, okay. I already have downloaded this. Wait, you're saying put this in the same directory as the GBK file?
Yeah.
Isn't this the GBK file?
Yeah, but this is a different GBK file. So this is from our genome, from the
Okay.
PROCA result. Yeah. Got it?
Yeah.
All right. All right, guys. So sorry. So now we can add our stuff in. Oh, first, you need to add your blast pad. So we downloaded it yesterday. Sorry, Jess, you also need to download this too.
I downloaded it on my desktop, so I don't have it on my laptop. I need to download it as well.
Okay. So you guys go ahead and download the blast. Jenny, you can just use the path we used for yesterday for A9.
Sorry, I need to
Download
download what also?
Blast. Yeah, so just share the screen again. Sorry, this is from yesterday's material.
No worries.
Yeah, I mean, this all gonna make sense if you watch yesterday's video later on. I think. For now, let me just help you out. So
Okay.
And we set the path and preferences.
yeah. So preference, break options. There should be a,
Last binary.
yeah. Make sure you select the folder up to the bin folder.
Which blast is it that we use?
Which blast?
Yeah, look at the bid. I closed it from yesterday. So I still have it down.
Okay, sure.
I don't remember.
Oh, you don't have to real.
Oh.
We already set that up. Yesterday. Just set the path up to the bin. So, download. Oh, so up to here. Okay, I have selected. All right.
Right.
Oh, okay. All right, so for this one, in right, so you need to select rig options and set the path for the blast. All right, good to go.
Thank you.
Okay, so now we're ready. Let's select the reference. So let's go to the directory where we had downloaded the GenBank file, which is for me, which is under this directory, big input. So I'm just gonna select this specific strain as a reference. So Okay.
Question mark?
Yeah. And then you can also select the query sequence folder, which is also in the same directory. Break input. I'm going to click OK. And after that, you can click on add to data pool. This should bring up all the GBK files under here. And you can also choose the output folder. I'm just gonna put it under my desktop and create a new folder. So you can click on next.
No options.
Yeah, you can just use the default options. Here, so you can insert how many rings you wanna generate. So since we have 3 genomes, we can add 3 rings. If you want like additional features added in, such as GC content and GC skew, you can add extra 2 layers here, but we're just gonna use the 3 gene bank file for now. So the top one, the most inner layer of the ring should be the reference, I think. So we can choose, you can click this one and change the name. With let's name it the strain name. So N16961. And then you can click on this gene bank file and you can add it to the ring. So now, oh, actually, I'm sorry. So you can click on this and you can click add data down here. So this, sorry about that. So you got to click on ring 1 first, and then click on the gene bank files and click on add data. Make sure your legend text indicate that you're on the ring 1. And for the ring 2, we're gonna add the Korean strain. This is the strain that is sampled from the environment. So we're gonna type in KORE1, and we're gonna also select the KORE1 and make sure you click the add data. And for ring 3, lastly, we're gonna choose our QCB genome from Illumina. So QCB.bc, and make sure you select the QCB.bc.gbk and make sure you add that to the ring. So once you have done all that, you can click on next. And you can also name your image title. I'm just going to name it QCB workshop. And once you're done, you can click on submit. And if everything's right, it should say it's running a blast. So while doing that, we can go ahead and check on our Rory output. So back to Hoffman 2.
Mine is still running.
Oh yeah. You mean Brick or Rory? Oh, yeah, pretty slow. How about yours, Jenny?
Is the output supposed to be Rory plots?
Rory out.
I have stuff in Rory now, but
Yeah, but it's still running,
stream
right?
is still writing into it.
Yeah, yeah.
I did get an error.
Oh, okay, okay. Kind of.
I guess they just simply
Oh, yeah, because the input path was different. Here we were meaning that the input file.
Oh, that one is just.
Yeah, yeah. So just need to do it. Yeah. So should be.
How about you, Jess? Is yours still running, the Rory?
I just added the things to the 3 rings. And now,
No, I mean in your Hoffman too.
oh no,
Do you have this
back from
output?
the beginning of class? Sorry, do I have what?
Is it still running, the Rory?
No,
Okay, I guess it's done then, right?
I got the Rory out.
Okay. Do you have the same content as mine?
A bunch of accessory and core genes and stuff. Yeah.
Yeah. And you also need this gene presence and absence .csv for
Yeah, I have that.
okay.
So in the brig, do I need to, I'm sure you said this, but do I need to do an image title before I click submit?
Yeah, I think so. Yeah. So that files will be saved under that name specific.
Right,
name.
okay.
Okay, so I guess for David and Jenny, for you guys, I'm gonna share this output file so we can proceed.
You just upload it to the drive.
Yeah, piece in the freezer.
Yeah.
So the gene presence absence .csv.
Yep. So Jenny, you can just copy and paste this. Are you in the Zoom? No?
No, I don't think it's in.
Okay. Let me just can you go to the Google Drive then?
In day 3.
Yeah, day 3. You should have, yeah. Should have downloaded and transferred it to me.
Yeah, so for Jess, for you, you can just use whatever stuff you have in the Rory output directory. So you guys can go ahead and install this in Hoffman too.
I was supposed to submit the blast, right?
This one?
Yeah.
Yeah.
Okay.
Click the blast and you should see the message saying running.
Yeah.
All right. So go ahead, type in this command. Oh, sorry. This one in your Hoffman 2. Just let me know if you guys are done installing this.
Mine finished.
Okay. So you guys can go ahead type in this command. So Python plots.py. So we're gonna put in our the SNP tree from yesterday, and then we're also gonna put gene presence absence data from the RORI out. So this should generate the three different PNG files. So you can go ahead and transfer it to your local computer once you're done. Yeah, so for now you can open up this one. So the pan genome matrix. So this clade is specific clade from yesterday's SNP tree. This specific clade has the Korean strain. As you can see from this matrix, you can see that there's a specific line that are shared among this clade. So this clade contains Philippine strains and the Korean strains. So this indicates that they share some of the genetic materials that other genome doesn't have. So in the paper, we actually found the genomic island specific to the Korean and the Philippine strains. So this is one way to find out whether certain genetic material is shared among specific genomes. Okay. Blast is still running. If you want to like speed up the break process, you can actually add the blast option. before you submit, actually, so you can actually submit. You can add the number of thread you want to use for your computer.
Oh, maybe I should have done that.
Yeah. It's still running, right?
Yeah, it's still running on mine. I'm assuming it's single threaded.
Yeah, that's why it's slow.
My laptop.
It would be a lot faster because it's only 3 genomes, but apparently it's not. Yeah, just for your reference, this is what the result will be look like. Since we didn't add the GC content and GC skew, you wouldn't have these 2 rings here, but pretty much you should have a ring shaped for the individual genomes that we put in. So here we can also see that some of these regions here is missing in Korean strains. Korean and QCB strains here, right? So this is one way to visualize certain genetic elements are present in one genome and not in the other genome. Okay. Yeah, and up to this point is pretty much everything for the workshop. So you guys, as Eloy said, you guys can use this QR code to evaluate the workshop. And we can still wait until this finished.
I'm gonna rerun it with more threads.
Okay. And for the homeworks, which I'm going to share with you guys tonight, let me just briefly explain what the homework is. Again, the due date for this homework is next Friday, as well as the quiz, which I'm gonna send out tonight. So the homework is, I'm gonna give this unknown FASTQ file. So you just need to assemble it and try to define what this genome is. Trying to define the species, what kind of species it is, using the ortho-ANI. So I'm gonna give you guys 3 different reference genome as a candidate. So I'm gonna give you a cholera genome, gonorrhea genome, and the E. coli genome. So you guys gotta guess which species it is. And also, I want you to guys run the contest 16 in the EasyBio CloudNet and extract a 16S RNA gene from that genome, assembled genome. And then afterwards, I want you to generate the break plot that we are running right now. You can just paste the image into this box here. So that's pretty straightforward, I guess.
I got some weird VM error when so this ran really quickly when I sent 8 threads. I got some weird VM error when I tried to render the image. It says it could not presume a space for much object heap. That looks like an OS issue.
What's the, how many memories do you have?
I have maybe this laptop with 8 gigabytes. Possible.
Maybe you can,
Maybe I'll try it again if it's 4. Maybe that.
sure, yeah, give it a shot. If it doesn't work, you can always use your desktop.
Yeah. My desktop is much stronger. I don't have the same issue. Yeah, I'll try this up too. Like, it's not really.
How to
Oh, so now you're selecting ring number 1, right? And you select this and add data. So yeah, this is somewhat confusing.
No, it's not. I just really did not.
Yeah.
It's like the first one.
Go back to the first one. Yeah, it is added.
Oh.
It's 600. Yeah, and the third one.
So it ran extremely fast and produced basically just a bear. So yeah, that was
Okay.
really fun.
Now, make sure you add the thread to the faster. This dash num.
Double
Visit our threads. Yeah.
So the homework assignment is due next Friday?
Yeah, next Friday.
Okay, and you'll send us the instructions?
Yeah.
Okay. Alright,
As well as the quiz.
okay. So that's also due Friday.
Yeah.
Okay. Alright, well, if that's it,
Yeah, sure.
so, yeah, thanks for everything.
Yeah. Thanks for joining though.
A lot of good